So hello, welcome back from the lunch break and welcome to this lecture series on bioanalytics. So we are two session chairs. My name is Petra Dittrich, I'm from ETH Zurich. And because we are two, we have to share our duties, and the first part of the duties were done by the other session chair.
So, hello everybody, my name is János Vörös, I'm also from ETH Zurich, and it's my pleasure to introduce the two speakers who told me that they will actually talk intermixed, so one or the other sometimes. So the first speaker is Orane Guillaume-Gentil. She studied pharmacy, and she's working in the Institute for Microbiology at ETH. And she's going to talk about why we need to analyze single cells and how to do this. And the other expert today is Professor Fredrik Höök, who studied physics and who is leading the Institute for Biological Physics at Chalmers Institute of Technology. And without further ado, I would like to give them the word so that they could introduce us how to do nanoanalytics with cells. Thank you.
Thank you. So good afternoon. As János introduced, so in this hour we want to discuss about, globally about the field of bioanalytics. What do we need bioanalytics for? What is the current state of the field? And also what are the challenges in this field, of course. And we would like to try to illustrate a little bit all these aspects in this presentation. And hopefully you will get a little bit of perspective from the side of the biology and also from the side of biophysics that we hope we can combine here. And so to start this hour, we would like to start with an example that we think is nicely illustrating all these different aspects of bioanalyticals. So from what do we need it, what are the challenges, what is it used for, and what could be the future direction in the field. And this example, this is one example we will hear much more about in the next talk, which is then the next generation drug delivery vehicles for biologicals. And so those biologicals are a very promising drug for next generation drug, let's say. But now instead of being small molecules, they are much larger molecules, biomolecules that we need to not only to bring to the target cells, to make them act on these targeted cells. We also need to make them enter these cells that they are targeted, of course. And so for this, there are a lot of current effort devoted to engineer nanocarriers for those biological drugs. And when designing such a new nanocarrier to achieve this task, there are several steps we have to have in mind and study and be able to control. So the first step is to assemble this nanocarrier, mostly based on supramolecular self-assembly. Then we need to control the molecular properties of our cargo, so being the size, the rigidity, stability, its content, and so on. Then we want this nanocarrier to be able to target specific cells and interact selectively with the type of cells that we are targeting. And then it should enter into this cell, so we need to study how it is taken up into the cell. And after this, we need the drug contained in the nanocarrier to be released inside the cell and reach its target. So we need, and after this, of course, we want to induce a cellular response in this targeted cell. And so those are a lot of different steps. All of them are really complex. And in order to design and construct an effective, such an effective vehicle, we now need different type of bioanalytics. So at first, we need to be able to characterize supramolecular nanoparticle assembly. We need to understand how those nanoparticles that we produce, how they will behave in a complex biological media, what will be their stability in such media. We also need to assess how it will selectively target and bind to the targeted cell, and later on how it will be internalized in such cells. Then we would like also to be able to probe what happens with the drug. Is it released inside the cell? Is it released in the sufficient concentration to produce the desired answer? And we also want to evaluate this biological response to the drug in the cells. So all of these steps really required advanced bioanalytics, and all of them, well, let's say all of them are really complex and acting at different scales and having different issues and challenges. No. So that's the first things, that we have a lot of different steps with different feature. And on top of that, we also need really advanced bioanalytics, because for each of these steps, we will not be able to obtain the necessary information with conventional analytical method, looking at average of hundreds of thousands of cells. So it's true for here the example of why going single cells and what it is needed in that case, but it's also true for going to single nanoparticle and single molecule analytics that we will see later on. So in the case of single cells, we now know that if you take a small piece of tissue from a human body, and then you look at the individual cells inside these tissue pieces, then we will find a high heterogeneity in between the cells that are composing this tissue. So all the cells mostly share the same genome, but then we will have different cell type, different cell states, and so on. But in addition to that, they're also organized in very defined spatial architecture within the tissue, where all the cells interact together to coordinate the entire function of the tissue or the organs. And on top of that, all this biological process are also dynamic. So the cells are also changing over time, or the properties are changing over time. And each individual cells will have also a different cellular response to different external stimuli or internal stimulus, with different amplitude or different rate of the cellular response. And so if you work with a conventional analytical tool and look at the average population, what you will get is a vague idea of what is the average behavior of those cells or characteristic of those cells. And from this, you would assume that all these cells are the same. So with bulk data, you completely mask this cellular heterogeneity, and then you're really limited in looking at any of these aspects that I just showed here. Now, going to single cell, single nanoparticle, and so on is one challenge. The other challenge is that we need those bioanalytical tools for really different scales. So if we start with the entire multicellular organism, like our cell, for instance, we know already quite a lot about the whole body behavior, but our body contains some 10 to the power 13 cells, some 20,000 genes that are common to all of these cells. And now these trillion cells organize into specific tissue organs that have a specific function. And those organs can be really complex too. So we take here the example of the brain. And in the brain, so in the body in general, we have about 200 different cell types. So they still have the same genome, but they don't express all the genome, and they have different transcriptome, which infer different proteome and different metabolism of the cells. And in the brain, for instance, we have 10 to the power of 11 cells that are coordinating an action together to provide the brain function. And just as an order of magnitude, just to give you a little bit the dimensions, so not for neurons here, but for an average human cells. So it typically speaks about some micrometer in physical dimension and some volume of in the picoliter range. Now, in another level, all those cells compose a defined tissue, but they also interact and communicate with each other through different pathways and using different molecules, of course. And so, typically in the brain, we have about 10 to the power of 14 neuronal connections. So there are really a lot of cell-cell communication in any of those tissue organs that we want to look at. And so it means that you don't only want to look at the composition or the cell composition of the different tissue, but you also want to look at the molecules, first the molecules that are inside the cell and that reflect what is this cell, what is its function, what is the state of the cell. And this is the basic type of molecule that we will find in the cell. So we have the DNA transcribed to the messenger RNA, which will be translated into proteins, which will then act on the metabolism of the cell. And if we take, for instance, the example of the messenger RNA that is inside the cell, so it's not only that you're interested in looking at this molecule in this complex mix with the other molecules, but within a single cell, you will also have to deal with very low absolute amount of the molecule you're looking at, which would reflect some few 100,000 of transcript within a cell. And among them, there are, depending on the cell type, some 10,000 different type of transcript that you may be interested in. And this plot here is just showing distribution of the abundance of different transcript species inside a human cell. And what we see is that there is a really broad distribution with a concentration on the copy number of a specific transcript inside a cell that can be as low as a few copies and as high as some 1,000 or even 10,000 copies of this transcript. So from the analytical point of view, it's of course very challenging to analyze those molecules. But now there is even another step or another scale. Bioanalytical is required, and it is to look at, for instance, in cell-cell communication, in small vesicles secreted from a cell to transmit some biomolecules. So take some that are inside the cell and bring them to another cells. And in those, you will again have some heterogeneity, differential content that you would like to be able to analyze. But this time we go down not to a few micrometer in size and a few picoliter, but to vesicles that have only 50 or 100 nanometer in size. And yeah, I give the word to Fredrik for more on those vesicles for a while.
Thank you. So yeah, I promise to continue from here, and this is because I have a great interest in solving the problem that Dave told us this morning is not solved, and that is targeted drug delivery. And while the communication across the synaptic cleft is fairly well understood, and the synaptic vesicles are not very heterogeneous, they are, but not anything compared with the extracellular vesicles that are released rather than the synaptic vesicles, they release the contact. Cells are also releasing extracellular vesicles, a small subfraction of which actually are released for a purpose. So the purpose is to reach neighboring cells or cells very far away to deliver genetic contact, to reprogramming these cells for functional reasons, but it is also involved in many disease states. And this shows that nature can communicate without the problems that we have when we develop the drug delivery particles ourselves, which is stimulating, and that is what in biomimetics is always stimulating us. But if we take a look at the cell membrane, we have approximately 300 to up to 1,000 different types of proteins, membrane proteins in the cell membrane, and 100 different types of lipids. And if you take the footprint of these molecules into consideration and then make up your dream or any nanoparticle vesicle out of a cell membrane, the possible heterogeneity is enormous. But it is also a smart design principle that nature somehow have learned to use. But in order to understand how cells have solved this issue, we need to analyze these particles in great detail. And that would be all right if they were all the same, but they are not. There are an enormous heterogeneity among the extracellular vesicles, which is illustrated here in this picture here. There is some kind of content, a genetic content. There are also other molecules and in particular you have a membrane enclosing the content of these vehicles containing of membrane proteins and very many different lipids. And to identify a unique functional subclass of extracellular vesicles, for example, exosomes, that are the ones that are believed to be responsible for this cellular communication, requires, of course, an understanding of these uptake steps, but you also need to identify them individually, be able to extract as much information as possible from a single nanoparticle. And that is very, very hard in complex molecular mixtures with low concentrations. But I will give some examples now on the existing methods, and then we will see how far we manage to go towards the end of the presentation and perhaps even present some of the latest development in the field. But these are the four perhaps most commonly used methods with respect to single molecule or single nanoparticle characterization. Electron microscopy, from which you gain information about size and structural information. Atomic force microscopy, size and to some extent rigidity. Resistive nanopore sensing, where vesicles or nanoparticles are transferred across a small nanopore in an ionic solution, and you probe the change in resistance across this pore. And nanoparticle tracking analysis. And I will come back to all these methods in more detail rather than going through this table here. If we start with electron microscopy and compare scanning electron microscopy with cryo-electron microscopy, the prime difference is in SEM, you need to immobilize the particles onto the surface. You need to dry, expose to vacuum, and the outcome of such analysis is that you get some kind of size, but this size is generally lower or obstructed by the preparation method. So the preferred method when it comes to nanoparticle characterization using electron microscopy is cryo-TEM, where instead you use a carbon grid and plunge freezing with as little water as possible from which you can extract both quite accurate size information and, in addition to that, also structural information on individual particle. I'm illustrating this here from, I think it is the second of these two papers. No, it is probably the first, where they have made a cryo-TM image of extracellular vesicles from a biological fluid. And by looking at the electron density and the structure, been able to characterize these extracellular vesicles into various subfraction structure-wise. But you don't learn so much from this. You only learn that they are different, not very much more than that. And I will come to that limitation soon, because you also would like to identify those that have specific biological markers, right? Atomic force microscopy is a technique that in principle would be able to do that. I'm just illustrating here what the type of information you can get from AFM, and that is that you get height information, and you can also check the rigidity of the particle. In this particular case, it was applied on viruses, where they show that by extracting or imposing a higher force, you can destroy them. With vesicles, you can learn or gain information about rigidity and to some extent size, but you need to take tip convolution effects into account and so forth. But this is a technique that is more and more used for also studying extracellular vesicles on an individual level, to look at this heterogeneity. But one interesting feature which is important to be aware of, if we compare this is just one example, but it is a quite typical example. It is that if we look at electron microscopy, which I had in the previous slide, the size is generally lower than that you get with cryo-TEM, and similar to what you get from liquid phase AFM. But in particular, in AFM, you often see reports that the height of the vesicles that you have characterized is really, really thin, from 4, 10 to 20. And this is because it is very hard to avoid tip effects on these soft objects. So this is something one needs to be aware of in operating with these techniques. This movie is supposed to be running live with these vesicles moving around in solution, detected in light scattering mode, where a laser is directed through a solution. And this is three-dimensional nanoparticle tracking analysis. And by following the motion of this particle in 3 dimensions, projected to 2 dimensions, you can deduce the diffusion constant, which relates to this size. And then you can get quite accurate size determination, again, of individual particles. But, and what one learned here is that a general conclusion is that NTA and DLS, which are both probing the hydrodynamic radius, give significantly larger sizes than both electron microscopy. That's easy to understand. It is less obvious that you usually have very large discrepancies actually between electron microscopy and DLS, because the hydrodynamic radius shouldn't vary as much as it often is observed to do. But this is so far only applied to define size, to some extent structure in using electron microscopy. But size is not the only thing we need to know about nanoparticle in order to identify, for instance, the subpopulation of extracellular vesicles from which we would like to learn enough to design drug delivery particles that really work. The ligand density and size combined is decisive for the fate of cellular uptake of nanoparticle, which is illustrated in a seminal work published almost 10 years ago now, which brings us now to the need of single-cell analysis in order to understand nanoparticle tracking analysis. And then after a few slides, now I will come back and discuss the few methods that have been developed in order to learn not only nanoparticle size and structure, but try to simultaneously determine more than one parameter, and in particular sort individual nanoparticle based on these features, and see where we are today with that goal. Welcome.
So back to the cellular level, so single-cell analysis. So I mentioned already that there are different types of molecules in a complex mixture with a wide dynamic range and so on. For fundamental studies in biology, for research in medicine or pharmacy, and for our example of drug delivery and whether or not the biomolecule has the desired effect on the cell we are targeting, we are now interested in analyzing the pool of intracellular molecule within single cells. And so for this part, I will focus on the transcript, so the messenger RNA. So looking at the pool of transcript will give us information about the functionality of the cell, about its response to the different extrinsic or intrinsic stimuli that it is encountering. Plus, it will have also some fluctuation due to the kinetics of burst of transcription followed by decay of those transcripts, which are adding noise in what we measure. But this is, at the time, the most advanced analytic part at the single-cell level. So, yeah, proteins and metabolites are much more challenging to look at because of various properties. But yeah, so I will just focus on the transcript so that we can see what are the different approaches that I use in current single-cell analysis. And so I will talk about three main different approaches. Here, one that is based on high-resolution microscopy of the transcript pool inside a cell. And then I will move to methods that are based on cell sorting, followed by RNA sequencing for looking at the entire transcriptome of a single cell. And at the end of our hour, I will come back and talk about an approach that we are exploring in our lab, and then some other groups are working on, where we now work on cellular biopsies instead of looking at the whole single cell. And so the line that I will follow here is what are the information. So I will stay quite superficial in those approaches, but look at what are the output and the respective challenge and limitation for these different approaches. And so starting with high-resolution microscopy, the idea is that the most common approach here is based on fluorescence in situ hybridization. But there are other fluorescent-based approaches. And then the general concept is basically to fix your cellular sample, permeabilize the cell, so you basically kill the cell, and then you can internalize a specific probe inside the cell, so you can target now multiple different transcripts inside the cell by introducing those specific probe for particular transcript, and then in that case you add some other component in order to fluorescently label those specific probe. And this allows you then to use high-resolution microscopy to image single transcript inside cells. And the interesting things in this approach is that while you kill the cell, you preserve their spatial relationship between the different cells. And you get a very high spatial resolution. So you can image individual transcript inside cells, and you get really a subcellular resolution with this analysis. And now there are a lot of effort going into this different type of approach currently. And it is now possible to really multiplex this imaging so that you are not imaging just one particular transcript or a set of 10, but now you can go up to more than 100 different transcripts within the cells, and this will probably explode to 1,000 or so in the next years. So it's not it's not a single transcript that you can look at. You can look at different transcripts, and you could do that with high throughput. So you can now image relatively wide field of vision with high resolution and image up to 40,000 cells now at once. But the limitation with this approach is that you need a predefined probe of sets, and so you are targeting known predefined transcript inside your cell. And in biology, what the trend is right now is not to look at something that we're expecting, but it's rather to compare as large amount of information we have about one cell, or cell under a certain condition, versus cells under other condition. Can be health and disease, can be with or without treatment, or different tissue and so on. And to do that, being restricted to known and predefined probe is really a strong limitation for the field. And another limitation towards the work that we are doing in our lab is that this approach requires you to fix and then permeabilize the cell. So you're basically killing your cells in your sample and cannot get any dynamic information about your cellular pool. So then the second approach I wanted to talk about is really the mainstream approach for transcript analysis in single cell nowadays. And this is basically an approach based in two steps. So in the first steps, you will isolate your individual cells, and then you will lyse the cell to access the endogenous molecule and perform RNA sequencing that had to be adapted from existing RNA sequencing for large cell population, but basically follow the same concept as we already used in biology before. And so in this first step, in order to isolate the cell, you will start with a tissue biopsy or two-dimensional cell culture. You will first need to dissociate, so to detach the cell from each other and from their eventual substrate. And then you will need to sort the cells in order to end up with individual cells before lysing it and accessing the molecules. And once you do that, so you have your lysed cells, what you can do is then take the whole pool of transcript. So we are not now just taking some selected transcript, but the entire transcriptome of the cell. You can reverse transcribe that to make a DNA library that you can sequence and get then the whole content in terms of transcript from the cell. And so back to those two steps, so there are now really many different protocols, both for isolating individual cells and for sequencing the transcriptome. But mostly, or initially, we used the fax to either just put individual cells in some wells. And now you can also add some labels if you have known biomarker on the cells. And you can also couple that to already pre-analyze and sort the cells as a function of some of their properties before doing the sequencing. And now, in the last years, this has been miniaturized into different formats. So you can have, instead of microwell, you have now nanowell reaction, where you can isolate individual cells in. You can use microfluidics to sort the cells in smaller volumes. And now you can even encapsulate individual cells with some beads and all the reagents for lysing the cells and initiating the library preparation in picoliter volume or nanoliter volumes. So the advantage of this approach in general is that this can be achieved really with a very high throughput. The initial approach and the microfluidic chips here are working in the order of hundreds of cells. But now with these microdroplet technologies, we can go up to tens of thousands of cells. So you can really massively parallelize your analysis and look at a large number of cells. And at the same time, we now really look at the whole transcriptome of those cells. So it's really a lot of information that can be gained with this approach. And the limitation here are that now you are not fixing and permeabilizing your cell, but you are removing the cell from its cellular context to isolate it and then lysing the cell. So you are also killing the cells and preventing the possibility to do dynamic analysis. Plus, you also disrupt the connection between the cells and lose the information about the context or the spatial and relative information of the cells that you are analyzing. So, yes, as I mentioned before, one of the limitations, so this is a great approach that has been massively used, and we gain a tremendous amount of information from those single-cell studies. But the main thing we learn is what are the cells composing a tissue and so on. And now a lot of efforts are put in order to add information and to add the information on how are these different type of cell or different state of cells related to each other in vivo in tissue. Are they neighboring? Are they communicating with each other and so on? And another aspect that I mentioned in the introduction was also this dynamic aspect that is very important in biology, where this step of lysing the cells at the end is also very critical because you have no way to monitor a single cells over time with this approach. So yes, indeed, sorting has the advantage that it can now be really achieved with this super high throughput and allows to now the direction is to make an atlas of all the cells in a human body, for instance. But there is this limitation of the lack of spatial and dynamic information. And so the approach we and other group are now proposing is that to change this background protocol, and now instead of this, to maintain the cells at the moment in 2-dimensional culture, later on and also in 3-dimensional tissue. And instead of going through all these steps, we will now use microfabricated probes to go inside a cell, take those cellular biopsy without killing the cells and without destroying the context, and then perform the analysis ex situ. So really preserving cellular context and cell viability. And there have been different approaches developed so far, using different type of nanoprobes, from nanoneedles, glass nanopipettes, and in our group we use this 3D FM that are hollow IFM probes. And so for time reason, I will just speed up just to show you in our work how this looks like. So we are using this 3D force microscopy. So our system, we have basically a miniaturized pipette where we can flow solution in and out of a probe with a pressure controller. And so at the end of our probe, we have a nanometer size aperture through which we can exchange the liquid. And as the probe is very small, we use here an atomic force microscope to drive the probe with force sensitivity relatively to the cell that we are addressing. And so it is now possible to take such a probe, target specific cells in tissue culture, insert the probe inside the cell, and apply under pressure to withdraw cellular content into our probe before lifting up this probe. And we can do that in a very gentle way so that we can preserve the cell viability by this manipulation. And there was some aspect that we didn't discuss today, but of course we had to have an efficient antifouling coating so that we can contact the cell without destroying it, and also flow the molecular content. And this is just a movie of now how it looks like. So we can visualize this extraction of cellular fluid in real time while extracting it. So in this example, we empty the cell. And so we can do that quantitatively, either taking up a few % of the cellular content or the entire cellular content. And just to show you that we do indeed preserve cell viability, it's just a movie where we extracted the lower cell but not the upper cell. And you see that both cells will behave similarly and divide nearly at the same time after this extraction. So it's really possible to take those cellular biopsy without killing the cells. And then, of course, we did, so this will be useless if we cannot analyze the content of those biopsies, so we did different type of molecular analyses. And very quickly here, so we could have an approach where we can extract, deliver the extract into some PCR mix, and then perform analysis of the transcript with here an example which you can see just a few extract analyzed for 2 different cell types, where we were able to get some 1,000 genes for some of these extracts, and looking at which genes are expressed in the different extract from the different cells, we are able to cluster differentially those different cell type and detect the differentially expressed genes between the cell type and distinguish them from each other. So reaching nearly the level that they are now with single cell analysis, it is now possible to do that also on small biopsies of cells that we can maintain in cellular context and preserve the viability of. So with this, I think we are over time already, and I want to close this presentation.
Can you check your microphone, please?
So thank you both speakers for giving us this great overview on the various analytical methods to analyze size or to analyze the content of cells, of particles. I'm sure there are many questions and we have time for questions, so please ask now. Okay.
Thanks, Orane, for a very interesting talk. My question, how many cells would you be able to analyze, let's say, in an hour, and which cells do you know you want to analyze? So which cells would you say are interesting? So would you need a second technique to basically pick the cells first, which ones you want to analyze, or?
Yeah, so for the cell we want to analyze at the moment, it's a really broad sort of application in pharmaceutical research, there are application in immunology, there are application in fundamental biology and so on. So right now we are still more on exploring what are the limits of the method we are developing and trying to make it suitable for as many cell types and so also adherent, non-adherent cells, cells within 3D tissue and so on. For the number of cells we can analyze, so the limitation here is that it is a serial process. So if you parallelize and so on, you lose all the resolution. So it will always be a serial process. And now the time it takes is really depending on what you do. So here, actually for the sequencing, I'm even preloading the probe with some RNase inhibitor, then going inside the cell. The extraction itself takes now about 1 or 2 minutes. But the time to transfer the substrate, to make all the picture of the cells, to preload, to take care of the sample after, and so on, I count like 15 minutes per cell or so. So I can do like 30, 40 cells a day. I can do several days in a row, but that's more or less the throughput. So now if you go to just inject something to a cell without doing anything more, then you could go to 2,000 cells a day. So yeah.
Thanks.
More questions? Over there.
So, up here. Thank you. Really enjoyed the presentation. For the cell analysis, not the nanoparticle part, CyTOF is often used now also as a means of providing a more in-depth characterization of cell types. So I wonder if you'd maybe comment for a minute on kind of how you see the complement between CyTOF and some of the single-cell analysis, CyTOF and some of the single-cell analysis you were just describing.
Yeah, so I'm a little bit biased towards this omics field and so on, and it's really this wish to look at what we don't know, so to look at differences that we don't know. So in my case, I'm not doing this because I need model system that we know what is happening to validate the method we develop. But I think the interesting here is really to be able to go for as much coverage as, for instance, the transcriptome or the proteome and so on. Now I'm also completely aware that all these methods, so both the optical imaging of molecules and mass cytometry, or all of these have also a tremendous potential. It's just like different application where you already know what you expect and look for it if it's here or not. And I think, yeah, both are complementary somewhat. And we are on our side just focusing more on the comprehensive molecular pool analysis.
Thank you.
More questions, come on.
Then maybe one to Fredrik. I was wondering, with these extracellular vesicles, we know that they can cause some diseases, you were hinting for that. But is it known how many and what types are the ones that actually cause these diseases?
No, there I'm not an expert, and there are probably others that can answer that question better in the audience here. But my understanding is that too little is known to really answer that question. But in order to eventually ask the question, we need to to characterize the heterogeneity with respect to subpopulations with distinct functions. Many of them have no function at all. It is just debris that is produced by the cells. But it is clear that they have a function. That, I think, is without doubt. But being able to identify what properties that are needed in order to have a function in disease or in health, I think that we are still waiting for that answer. And of course, we need to be able to identify them sufficiently well in order to sort them in sufficiently clean samples to maybe apply ensemble averaging method at some point, where with higher concentration you can gain much more information than you can with a single. So I think the sorting step, that is the, if you're interested in nanoparticles and heterogeneity in biological nanoparticle, the sorting step, I think, is the step to put effort into right now.
I have a question related to the sorting step. You showed this vision of a sorter for this nano sorter. I didn't get it right, but so your microparticles or vesicles are immobilized on a membrane, right? Mm-hmm. And then you can see by flow, you can see how they would move. So, but then you have to sort them. So you want to apply an electric field in order to.
The idea we have is an electric field, and we are moving away from having them tethered to the surface. And Quentin in the audience is actually, or have been working, or are working on a solution where they are instead free in solution. Yeah. Because that is a requirement. But then you need to introduce a drag, because if you don't have a drag, they will move with the speed of the flow, irrespective of which size they have. Mm. So you need some counterforce, and then you need to know that counterforce in order to extract the size. Mm. But maybe the difference in size is enough if you calibrate them, because that theory is not straightforward, unfortunately.
Sounds like a challenging project.
But we are now a couple of talented students addressing a particular
good luck in finding one. Do we have more questions? Yeah, there is another one.
And so I have a question for you. It's about the heterogeneity. Most of the time, the technique used is the fluorescence, right?
Once again?
About the heterogeneity.
Yeah.
People are using the fluorescence, right, to see differences in the populations. But do you think that it's the best technique?
No, electron microscopy in solution with atomic resolution would be the best. No, it's. I think it is better than everything else right now. I actually think a label is better than you could imagine scattering holography, something like that. But the resolution is not really there to generate strong contrast. So I think we will have to work with labels for yet. At least I haven't seen something that beats that. Mm-hmm.
I have many questions, if you, but I would like you to ask some.
So I'm curious about this biopsying compartments of cells or volumes inside cells. So coming back to compartments, can you sample mitochondria or organelles? I mean, I guess it depends on the hole you use in your cantilever, in your tip.
Yeah.
And the suction.
So in this initial work, we worked with it's a triangle with a 400 nanometer size, and it's basically a molecular sieve. So we harvest whatever is soluble and small enough to go through this aperture.
So if you sample the same cell, how different are these samples?
So it's hard to say now, because this work on RNA sequencing is the first, well, it's ongoing now. And that will be the first analysis we do downstream that would allow us to get some resolution to start to look at this. So how reflective is this biopsy from the whole cellular pool? So what is at all? So we know we don't kill the cell, but for sure there is a perturbation. And the cell will react to this depletion. So what is this response that we will need to characterize if you want to go for dynamic studies? And we are just reaching now the means to look into that, at least at the transcript level, with this type of analysis. And before we had only like proof of concept studies that tell us we do take this molecule, we do redispense it, we are able to analyze it. But we don't have enough information to look into details into that. And just back to this question of what we can target. So here we can go inside the nucleus or the cytoplasm, and now we are working on this. So we managed to have larger aperture that can still be inside the cell, where we can extract mitochondria. It's also ongoing work, so at the moment we can harvest maybe 20, 30 mitochondria. It's much more local now, so we see only around the tips we take them, and we can then retransplant them and re-inject them in other cells and so on. But it's so we know it's feasible, but how feasible, it's an ongoing process. So.
Thank you.
Good. If we have no more questions, let's thank our speakers again. Thank you.